Good morning, everyone. Thanks for joining. It's my great honor to introduce our guest speaker, Dr. Jim Hurley today. Dr. Hurley is the professor of Department of Molecular and Cell Biology at UC Berkeley, and he was elected as a member of the National Academy of Sciences early this year. Professor Hurley achieved his PhD degree of biophysics from UCSF. Then he moved on to get his postdoc training at the University of Oregon. In 1992, he was appointed as the tenure-track investigator at the NIH and was promoted as chief of section on structural biology and cell cycling years later. After 20 years career at NIH, James decided to move back to West Coast and establish his lab at UC Berkeley in 2013. His research groups always interested in fundamental questions about how the interactions between proteins and membranes determine certain organelle shape and the evolution of shape over time, and how protein membrane interactions regulate the cellulose during the essential cell processes. Specifically, his research reshapes our understanding about gas called lysosome system, autophagy, and cargo sorting into coated vasculature, especially about how pathogens subvert and co-opt these systems. On the other hand, in Dan's group over here, we identified HPV as the first viral cargo of one of the coated systems, retromer. So it's not surprising that we noticed Jim's fantastic work, especially because his high resolution structure of retromer complex and proposed its function architecture. So now his research even attracts more attention from us when we are moving our research towards GTPase activation protein regulation, HPV retromer interaction, and also how HPV prevents lysosomal delivery. Another thing I'd like to mention is James extended his research into the neurodegenerative disorder field because both autophagy and lysosomes are attracting increasing interest as treatment targets for these diseases. So, all right, please help me welcome Dr. Hurley. If you have questions, please put them in the chat box after the presentation. James.
Right, thank you so much, John, for the invitation. Right, already got ahead of myself with the slides. So this is my first full day remote seminar. So it's quite exciting. I decided to talk about what I'm most excited about in terms of the recent results in the lab. It perhaps wasn't quite what John had in mind when he invited me, but I hope it's not too dull. I'm going to talk about regulatory mechanisms in lysosomes and autophagy. And the cartoon on my opening slide shows a growing phagophore that's in the process of engulfing cargo, closes into an autophagosome, fuses with a lysosome to form an autolysosome, and eventually back to a lysosome. And the lysosome itself regulates autophagy. So I'm going to begin the talk with a couple of stories about regulation at the lysosome and how that feeds into autophagy. And then in the second half of my talk, I will tell you more about the process of autophagy itself. Story number one has to do with the lysosome as a signaling hub. And this is an area that I got interested in after moving to Berkeley. Roberto Zonco moved to Berkeley 6 months after I did. And we formed a partnership to study lysosomal signaling mechanisms, which really was a new area for me at Berkeley, and it's been very rewarding. This cartoon shows kind of schematic of the lysosome as a hub in which inputs such as energy, amino acids, growth factors, oxygen are integrated, and outbound signaling regulates things like autophagy, lysosome biogenesis, protein synthesis, lipid synthesis, nucleotide synthesis, and all of this signaling is coordinated through the mTORC1 kinase complex. And so this kinase won't actually appear explicitly anywhere in my talk, but it's always in the background. So mTORC1 is active on lysosomes in the presence of pro-growth signals, such as abundant amino acids. It's inactive typically on the cytosol, so in conditions of starvation, stress, the absence of growth factors, low energy, it will be in the cytosol, it will be inactive. And so let's focus for a minute on how the interconversion between active mTORC1 and inactive mTORC1 occurs. There's a 5-subunit lysosomal complex called regulator. It consists of lambtor 1 through 5, for those of you who are specialists, which binds to 2 GTPases of the Rag subfamily and is functional on the lysosomal membrane. So this regulator Rag complex is central to mTORC1 regulation and central to the talk. Now, the Rag GTPases are subject to regulation by GAPs, GTPase activating proteins. There are 2 major GAP complexes, GATOR1 and folliculin FNIP, with antagonistic activities. The folliculin FNIP complex promotes mTORC1 activity, while GATOR1 antagonizes mTORC1 activity. Just to go a little deeper into the roles of these GTPases, the RAGs are functional again as dimers, which sets them apart from almost all other small GTPases, and consist of 1 copy of Rag-A or Rag-B and 1 copy of Rag-C or D. And I will refer to these really just as Rag-A and just as Rag-C for the rest of the talk. In fact, our experiments use A and C, but if you switched B or D for A or C, you would I think, get the same results. Now, the active Rag regulator complex contains a GTP bound to the Rag-A subunit and a GDP bound to the C subunit. So in a more typical situation with a small G protein, the GTP form is active, the GDP form is inactive. With the RAGs, both are important. So the correct nucleotide state is required in both cases for full activity. The reciprocal situation with GDP on Rag-A and GTP on C is inactive. So this does not recruit or activate mTORC1. So this conversion between the 2 is really the central event. GATOR1, in the presence of low amino acids, pushes this reaction to the right on this diagram, and folliculin FNIP pushes it to the left. So folliculin's GAP activity promotes activation in the presence of high amino acids. GATOR1 does the reverse, and we were interested in how this occurs. So there's a bit of a paradox here, and I'm going to give a shout out to Sean Ferguson, who I saw as among the participants. And in 2013, he published a paper showing that folliculin localizes to lysosomes in starvation. But I just told you that folliculin is responsible for the activation of mTORC1 upon feeding. However, this shows that on feeding, folliculin is released from the lysosome. So what on earth is going on? Yeah, what I like about the story I'm about to show you is that very often structural biology is a form of photojournalism. We provide beautiful, detailed pictures of events that we basically understand. So we provide a lot more detail on what at say the genetic or cell biological or physiological level people feel they know. We might provide a lot of structural complexity that had not been anticipated, but very often this complexity does not fundamentally change how we think about the cellular events. And I'd argue that the story I'm about to show you really does change how we think about things. And I think that's true for actually all of what I'm going to show you in this lysosomal part of the talk, and that's why I'm kind of so into this story right now. So what we discovered, and we is Rosalie Lawrence, a graduate student in the Zonku lab, who worked very, very closely with my group, is that if you simply purify folliculin FNIP, we're working with FNIP2, and mix it with Rag and Regulator in the inactive and active states. What you see cartooned here is the active and the inactive state. It forms a very tight complex with the inactive state, and it inhibits its own GAP activity. So in this size exclusion chromatogram, what we're showing is a stable complex with the 5 subunits of regulator, the 2 rags, folliculin and FNIP, formed with the nucleotides in the inactive state. And we are, by the way, using an XTP specific mutant of Rag-C. So we're able to separately control the nucleotide state of both of the rags in all of the experiments that I'm showing you. And throughout the study, we use HPLC of the nucleotides to get an accurate idea for what is bound. So this turned out to be a very nice substrate for cryo-EM, and Simon Fromm, a postdoc in my group, was able to get cryo-electron microscopy structure of this to 3.6 angstroms. It's a very elongated structure. You see here the Rag dimer. So here's Rag-C bound to XTP, Rag-A bound to GDP, the 5 subunit regulator. And here you see this elongated folliculin FNIP complex. The long domains of folliculin and FNIP are protruding into the cleft between the Rag-A and the Rag-C. And you'll see a theme here that the longan domains I will show you have GAP activity, and the cleft between Rag-A and Rag-C is going to be home to all sorts of interesting action. Many different proteins turned out to bind there. So in order to dig deeper into the mechanism, we had to know what the catalytic residue is. That was not known, but we were able to infer it from work on the Gator-1 complex from the Sabatini group. They had solved the structure of NPLR2 as part of the Gator complex and localized the arginine finger required for GAP catalysis to arginine 78. And we inferred that arginine 164 of folliculin would be active. And then we were able to show here that the R164A mutant completely lost GAP activity. So we now know where the catalytic site is. And when we looked at the structure that Simon had just determined, we had a nicely localized XTP density in Rag-C, and the arginine responsible for catalysis was 23 angstroms away, so much too far away to be active. So this told us that what we saw was an inactive complex, a very stable and completely inactive complex. So we named it the Lysosomal Folliculin complex because this is the state responsible for the stable and inactive form that is localized to lysosomes. So what is this good for in terms of regulation? I'm going to turn now to the master regulator of lysosome biogenesis, TFEB. It's an archetype of a larger family of lysosomal transcription factors. These regulate the transcription of proteins needed for autophagy. They regulate the transcription of the factors needed for lysosome biogenesis and lysosome function, and they're important in a wide range of diseases shown here, neurodegenerative and otherwise. And what I want to emphasize, what you need to know to understand the upcoming slides, is that in amino acid fed cells, when the lysosome is replete with amino acids, TFEB is phosphorylated, it's inactive and cytosolic. And the role of the folliculin GAP activity is to support this amino acid fed state. So when the folliculin GAP is active, Rag-C will go to the GDP state that promotes active form of the Rag dimer that promotes mTORC1 activity, mTORC1 phosphorylates TFEB. It is retained in the cytosol and inactive in transcription. In amino acid starvation, all of this stops. TFEB is dephosphorylated, it's active, and it's localized to the nucleus. So the lysosome folliculin complex that we've just discovered in this model, we now predict would support the amino acid starved state. So let's see if that's true. I told you that the ARG164 mutant is inactive as a GAP, and the consequence of that is that you can drive TFEB, or TFE3 in this case, to the nucleus even under fed conditions in a highly statistically significant manner. So this is an experiment done by Rosalie where she's used shRNA to knock down folliculin and then carried out rescues with either wild type or 164. And you can see the dramatic shift in the cytoplasmic to nuclear ratio consistent with the prediction. So we have a point mutant to knock out the GAP. We wanted a point mutant to knock out the LFC. We went back to the structure and examined the contacts and found that particular phenylalanine 118 form tight contacts. We mutated it to an aspartate, and that blows up the complex in vitro. When that is put back into the cells in the same RNAi rescue experiment, the results are perhaps not, it's not an all or nothing result the way we saw with the GAP, but it's highly statistically significant and consistent with the model that even in starvation, much of the TFE3 is retained in the cytosol, which is not the case with wild type. So this now begs the question, what is regulating the lysosomal folliculin complex? So we've discovered this complex. It explains how folliculin can be shut off in starvation, what turns it back on. Now, a literature going back a few years had implicated a lysosomal arginine transporter called SLC38A9 as the activator, and it had been proposed that it might be a GEF, which would promote the acquisition of GTP by Rag8a. Now, we had observed that Rag8a spontaneously acquires GTP and went and looked at this in a little more depth. I should tell you, SLC38A9 is an 11 transmembrane crossing transporter, and it has an N-terminal cytoplasmic extension that communicates with the signaling machinery outside the cell. This was already known, and it was known that it is released on arginine binding. So in amino acid replete cells, the cytosolic extension is free for binding. In starved cells, the cytosolic extension is sequestered. So what we did is made a construct where we chopped that tail off. We found that made a very stable complex with regulator, that is the lambtors, and the Rag8a dimer, and these are in the inactive state. So this turns out to support GAP activity. You can prepare the folliculin complex, mix the SLC fragment, and restore the hydrolysis of XTP. So undetectable here, indicating that the hydrolysis has consumed all of the reactant. And we determined a cryo-EM structure of that. We is Simon Fromm. And the SLC binds right between the cleft between Rag8a and Rag8c, just where the tip of the folliculin FNP complex bound. And if we look in detail at the structures, we can see that when you go between the active and inactive Rag dimers, there's a large structural change. It's about an 18 angstrom widening of the gap. That explains why SLC doesn't bind to the active. The gap is too wide and there's not a snug fit. Conversely, in the inactive state, there's a nice snug fit for the SLC helix, and it sits right here. There's very little change in the geometry as compared to folliculin. So what this suggests is that the SLC directly competes with the binding of folliculin FNIP in that LFC. So in order to determine if that's true, we carried out an incubation where we'd assemble the LFC and then add the SLC fragment, leave it overnight and run it on gel filtration. This figure is a bit complicated, but the upshot is that after that incubation, the complex completely falls apart and all that one gets are the separate components. This is the model now for how 2 steps in the reaction work. You would start out in the starved state with this Lysosomal Folliculin complex. It's inactive as a GAP. So it's not passively inactive. It actually, in a kind of proactive and specific way, it is preventing GAP activity by very, very stably picking the sites that to be accessed, the folliculin side, Rag side. When cells are refed, the cytosolic tail of SLC38A9 is released, and that is able to bind to the rags in such a way that it kicks the lysosomal folliculin complex apart, but it does not block the GAP conformation. You then get the gapping of the Rag-C, you get activation of mTORC1, and a nuance of this is that it is activated specifically toward TFEB. That's a kind of detail that's worked from others that I'm not going to get into. Let me tell you my second story about lysosomal complexes. And this is fitting perhaps because I was invited by the genetics department. I haven't said anything about genes, but C9orf72 is a gene which is important in neurodegeneration because it is the gene that is most commonly mutated in both ALS and FTD, 2 serious and incurable diseases. So what does this have to do with lysosomes and with what I showed you? So a couple of things go in these C9orf72 mutants. There is a hexanucleotide repeat expansion. So GGGGCC is expanded in an intron in a non-coding region in C9orf72, and there is a gain of a function effect where the RNA repeats and dipeptides encoded through a non-ATG dependent translation mechanism aggregate and are toxic to neurons. That at the same time, there's a loss of function where this the presence of the repeats lead to a reduction in the expression of the C9orf72 protein. So what does the C9orf72 protein do? There are a number of ideas coming out of studies of engineered mice that point to reductions in autophagy, for example, motor impairment, hippocampal neurodegeneration. These correlate with the presence of the dipeptide aggregates, but it's been shown that they could be alleviated by re-expression of the full amounts of wild-type C9orf72 proteins. So there is a synergy between the loss and the gain of function, and we were interested in better understanding what is the normal function of C9orf72 that is lost. So a number of labs, 7 labs, including Ferguson lab, this paper by Amick and MBC, discovered that as part of its normal role, C9orf72 forms stable complex with 2 other proteins, SMCR8 and WDR41. Not much was known about this complex, but there are 2 longin den modules, C9 ORF72 itself and SMCR8, both contain den domains, and work by a number of people, including Karen Reinisch, also on the call, had shown that other den domain proteins are GAFs, so guanine nucleotide exchange factors. And it had been proposed that this complex was a GAF for Rab8a and Rab39b. So I will come back to that. But apart from this idea about it being a GAF, very little was known about the normal function. So Mingyuan Su in my lab determined the cryo-EM structure of this. So again, without really knowing what its normal function was or what we were looking for, she went out and got a picture of it, and she compares it to an islip hook. Let's see if that, no, the picture doesn't come up, but it's a little bit like an eye and a hook. That didn't help us much with the function, but this did. We compared it to the folliculin structure we just solved, and it turned out that the overall geometry of the longin domains was the same. I mean, and clearly this was part of the motivation in looking at it at the first place. It fit with the theme. So after having wondered for a long time what the arginine finger was, we realized that all along we could have simply read it from the sequence. If you read the sequence, it's ARGF. So arginine finger, and this ARG in the middle of the ARGF motif is the arginine finger. So we mutated this, but we didn't know the substrate. But first, I want to return to the issue of the GAF. Now, it's perfectly possible this could be both a GAP and a GAF, but what I've just suggested to you is that it's a GAP. Is it a GAF also? It's not a GAF for Rab8a and Rab39b. What I'm showing you here is a control experiment with Rab5 and the well-known Rab5 GAF Rab8a. So this shows you the drop in the MANT GDP fluorescence you should get as you replace GDP with GTP. So this is what functional GAF will look like. And we had absolutely no change in the MANT GDP. So there is no catalyzed GDP release by the C9 ORF72 complex for either of these two small G proteins. So we believe that this is a GAP, and one of the great things about Ming, not only does she work extremely hard and know cryo-EM very well, but she also reads the literature exhaustively and really thinks about the literature in cell biology and neuroscience, which is not true, I think, in students in structural biology. So for any of you, if there are any structural biologists out there in training, the lesson is read every paper you can, as far out in physiology as you can get. So don't ignore it because it's not structural. So Ming ran across a paper in Nature Neuroscience that reported that knockdown of ARF6 rescued defects in actin dynamics in iPSCs or iPSC-derived motor neurons from C9 ORF72 mutated ALS patients. And so she had the idea perhaps could be a GAP for ARF proteins. And I should say that she tried a large number of different small G proteins, so this and got many negatives. So she confirmed that in this fluorescence-based assay, she got the characteristic drop in the presence of wild-type ARF1 and the C9 ORF72 complex, and this was replicated for ARF6. They behave the same, so it appears to be an active GAP with respect to ARF1 and ARF6. If you try this with folliculin FNP as the GAP, you get absolutely no reaction, so it is specific. If you try it with the Gator complex that I mentioned, the Rag8a GAP, you also get no effect. You do not need the WDR41. Sean Ferguson had shown that WDR41 is responsible for lysosomal recruitment, which we find as well, but it is not needed for the GAP activity. This makes sense because the arginine is at the opposite end of the WDR41 binding site. This is also true for ARF6. Now, if we use the GTP hydrolysis deficient ARF1 or ARF6, there is no reaction at all. So this depends on the canonical GTP binding site in the ARFs. If you use wild type ARFs, but you use the arginine finger mutation, this is also dead. So it is consistent with what we know about the behavior of this type of GAP. We can really beat this to death and look at this by the HPLC assay as well. So we now here we'll set out the reaction. We'll have a standard where we know where the nucleotides run. A GTPase will start out with some ratio, some in the GTP state. Usually there will be some trace GDP. If the reaction is effective, we'll see the total conversion of the GDP peak to the GTP to the GDP peak. So when we carry out this experiment, here are the starting states. Now watch the peaks. We'd expect the GDP peak to increase and the GTP peak to disappear for an active complex. And here you see in the wild type complex, you have complete conversion. The GTP is eliminated. It all goes to GDP, and all of the controls, you see no change in these HPLC traces. Now, another paper came out. We were not the only ones to solve the cryostructure. This is actually from a former postdoc from my group who moved to China and was working on this completely mutually unaware. And he had proposed that Rab8 and Rab11a were the substrates, and we went back and compared, and the activity is non-zero for Rab8a, but the conversion is not complete. So the activity in our hands is far stronger with the ARF proteins than the Rab proteins, although I would say the Rab8a is the only thing other than ARFs that has non-zero activity. Even the ARL proteins, the lysosomal ARF-related proteins, have zero activity in our hands. And so what do we think is going on? This is the model for what we think is happening in a normal cell with normal levels of C9orf72 mRNA and protein. This protein will be cycling on and off the lysosome depending on the nutrient state and converting ARF GTP to ARF GDP. Some GAF, the identity of which is unknown, reverses this process, and ARF GTP is involved in many different events in membrane trafficking, organelle biogenesis and homeostasis, signaling and cytoskeletal reaction or regulation. And now when C9orf72 is haploinsufficient, the idea is you build up an excess of ARF GTP and you have less ARF GDP. You get excessive signaling in various processes. And somehow that excessive signaling decreases the autophagic flux and increases the toxicity of the repeats. I think how that happens, which of the specific events in membrane trafficking, the cytoskeleton and so on, are responsible for the drop in autophagy and the increase in toxicity is a burning question. Another burning question, obviously, is what is the identity of the GAF or GAFs that antagonize C9orf72? Because I think it's pretty obvious if this is out of balance, if you have a corresponding inhibition of the appropriate GAF, you could perhaps restore the balance, and that could be a therapeutic, and clearly we're excited about this and pursuing this possibility. Now, I had mentioned autophagy, and I'd like to spend the rest of my talk talking about autophagy. So I'm going to change gears, and you'll see in the upper left, there is now a little autophagosome to remind you that we have just transitioned. So autophagy is really important as a basic cellular mechanism, and in terms of disease because of its role in clearing protein aggregates, clearing intracellular pathogens, and in the removal of damaged mitochondria, among just to cite the most famous of many other processes. And the uptake of specific cargoes like aggregates, pathogens in mitochondria is referred to as selective autophagy. And up to only a little more than a year ago, the paradigm had been that cargo was engulfed by pre-existing autophagic membranes. And the autophagic membrane was was designated by the presence of LC3 conjugated to the surface. LC3 is a ubiquitin-like modifier that is linked covalently to the membrane of the autophagosome. A series of 3 papers from the Ewell, Rando, and Martin's labs, and we were part of the Martin study, in the spring of 2019 really led to a shift in the paradigm and led to the idea that these membranes are created on a just-in-time basis in the presence of cargo. So the idea now in the new model is that cargo itself is the trigger for the formation of the membrane. And then we set out to look at the mechanism of this, both using structural biology and reconstitution. Just to highlight the main players, there's a kinase complex called the ULK1 complex, a lipid kinase complex containing VPS34, the PI3 kinase complex 1, a system consisting of the ATG 12, 5, and 16 proteins that functions in the same sort of role as an E3 ubiquitin ligase in this conjugation reaction. The WIPI proteins, which are beta propellers that bind to the membrane and bind to the lipid product of the PI3 kinase complex. And then finally, I'm not going to have time to talk about these, but transmembrane protein called ATG9 and a lipid transporter complex, the ATG2 WIPI4. And these work with cargo adapters whose job is to recognize substrates. And the 2 that I'm going to be describing will be NDP52 and optineurin. And we picked these 2 because of their role in mitophagy and the uptake of mitochondria. So I'm just going to tell you a little bit about the structure of one of these complexes before I plunge into the biochemistry and what they actually do. And a FIP200 is the scaffolding subunit that holds together the ULK1 complex, and we really think it is responsible for organizing the overall shape of the autophagy initiation complex. And Adam Yoakum, a postdoc in the lab, showed that this forms a large C-shaped dimer. It's a very extended, it's made it hard to align and hard to get good resolution, but the shape is quite fascinating. And despite all the flexibility, he was able to get a 9 angstrom or so structure. It really came to the surprising conclusion that this looks like another protein involved in autophagy regulation called TBK1. And this was really not something that we had expected to see. This gives you an idea of organization of this ULK1 complex, which is so central in autophagy regulation. In addition to the C shape, there's really a great deal of intrinsic disorder. So there are 2 flexible regions that let these other units, the ULK1 kinase domain and the ATG13-101 subcomplex, float about on long flexible tethers. So this seems to be an unusual complex in that there is so much intrinsic disorder. The entire FIP200 molecule goes far beyond this C-shaped dimer. So this N-terminal domain is the C-shaped dimer, but there's a very long coiled coil that protrudes from this. So this is a histogram of distances taken by negative stain EM, but it's up to 105 nanometers contour length, or 95 end to end. And this region at the tip we call the claw domain, and this is responsible for binding some of the cargo adapters. So this is a really nifty structure with this curved arm that is organizational and holds the complex together, and then a coiled coil that can extend nearly 100 nanometers to engage with cargo and cargo adapters on its far end. So that's the briefest account of the structures I think I've ever given, because I wanted to leave time to plunge into what they do. And one thing that this does, we've studied by hydrogen deuterium exchange, which is a mass spec technique that's good for probing conformational changes. And we've shown that when cargo engages at the C terminus, it provokes a kind of opening up of the hydrophobic parts of the coiled coil that increase its affinity for membranes. So why does this matter? I'm now going to segue to the meat of the autophagy story. And this concerns a reconstitution. And so in a cooperation with Sasha Martens, who is at Max Perutz Laboratories in Vienna and does similar work to me, we decided instead of competing, as would be typical for labs doing similar things, we would join forces because there were so many proteins that needed expressing and purifying. And so we have now purified all these components that cover this autophagy process, not the whole of autophagy. LC3 lipidation is not autophagy, but nevertheless, this is the majority of the factors that are involved in the pathway. And we are using as a membrane source giant unilamellar vesicles, which are cell-sized vesicles. And these are supposed to mimic the membrane source in autophagy. And so this is the lipid composition. We picked something that was, we're trying to be fairly close to the ER, but we also had to leave plenty of PI for the PI3 kinase, and we had to leave plenty of PE for the LC3 lipidation. So this is an example of one of Chunmei's experiments. In this case, what she's done is just taken, as a cargo model, she has GST dimers fused to tetraubiquitin, and then she's taken the adapter NDP52 and showed that in the presence of cargo, that the ULK1 complex is strongly recruited to membranes. And this is something we expected based on that hydrogen deuterium exchange that I showed you, that shows that the ULK1 complex goes through a allosteric transition that makes it more friendly, more prone to localize to membranes. So in another study that Chunmei did, this one was with the Martens lab using the LC3, the human LC3 conjugation system they developed. We are now looking in the presence and absence of WIPI2. WIPI2 is kind of major scaffold of autophagy initiation. We can see strong recruitment of this E3-like complex. Again, the E3 refers to ATG16-12-5. And we get a nice in vitro conjugation of LC3. And I should say we've done extensive controls using the protease ATG4, using FRAP and other techniques to show that the LC3 is covalently conjugated to the lipids. It's not reversibly bound. So those two things, I've just given you vignettes of recently published work, and now what I'm going to show, we've put the system together from start to finish. And so what you're seeing here is we're starting with a core mixture consisting of WIPI2 and the LC3 conjugation machinery. And here in the E3 channel, we're looking at recruitment of the ATG16-12-5 complex. And here in the LC3B channel, we're looking at the LC3 conjugation onto the GUV. When you add the PI3 kinase, you get a substantial boost. You didn't see much without it. The presence of ULK1 and C1 is similar. You don't see an enormous boost. The adapters alone don't do much. Very little LC3 lipidation. The activity with the ULK1 alone is a little disappointing. We get some significant stimulation of the C1 by the adapter and cargo, but finally when we put it all together, we get the robust and fastest recruitment. And here what I'm showing you are the kinetics done by an unbiased integration across many fields of view of GUVs. The error bars are large because GUVs are heterogeneous, as any of you who work on this I think well know. So this shows you we get a large stimulation with the C1 PI3 kinase complex. We get a larger stimulation with ubiquitinated cargo, the NDP52 adapter in C1. And we get the largest effect when we combine the whole system, which we'd expected. So not only is the maximal signal large, but the velocity of the reaction is faster. Now, a second adapter, optineurin, is considered even more important in mitophagy and is implicated in neurodegeneration. So we took a look at that. Optineurin is known to be phosphoregulated by the kinase TBK1. So in the experiments I'm showing you, we are using a constitutively activated optineurin mutant. And so let's see what happens here. Again, the C1 complex and ubiquitin potently stimulate. There is a big difference, however, the ULK1 complex does not. In the presence of the C1, the ULK1 is slightly, probably not statistically significantly, less active. Now, this fits with the biology. A recent publication in JCB showed that optineurin, unlike NDP52 dependent mitophagy, is independent of FIP200, a subunit of the ULK1. So what we're seeing, I think, would be surprising if we hadn't done that in the wake of a paper that found something quite similar in cells. So now if we look, instead of looking at the recruitment of LC3 or the recruitment and covalent lipidation of LC3 and the recruitment of the E3, if we look at the recruitment of the ULK1 and C1 complexes, so we've now flipped the script, we're looking at recruitment of these more upstream elements, we see that the NDP52 recruits or is recruited by the, misspoke. The ULK1 complex is recruited by NDP52, but not by optineurin, just as the downstream effects would suggest. And when we flip this around, we can see that the C1 complex is strongly recruited by optineurin and somewhat less strongly recruited by NDP52. Again, consistent with what I just showed you. So this picture seems to tie together quite neatly. So in summary, this is, we think, a powerful assay for dissecting autophagy mechanisms and looking at properties that we can look at with all the levels of control you have in an in vitro system. It's been a long road to put all of this together. And in terms of the specific findings of this set of experiments, we really come to see autophagy initiation as robust and is mediated by a web of many interactions. So early portrayals coming out of yeast genetics had led to the model that autophagy initiation proceeded in a very linear manner with ULK1 kinase second and so on. What we see instead is that there's a web of interactions, and moreover that web is put to use in different ways by different cargoes. So depending on whether you are, say, recognizing your damaged mitochondrion with NDP52 versus optineurin, the nuances of which subset of the core machinery are used are different. And I think I'm finishing just about on time. So I'd like to thank the folks who did the work. there are really five people in the lab currently who did the work that I focused on. Simon From did all the cryo-EM in the Rag regulator system. Ming Yuan Su did all of the C9orf72 work. Chunmei Chang does the autophagy reconstitution. Shan Shi, the hydrogen deuterium exchange and biochemistry of the OLK1 complex, and Adam Yoakum, the structural biology of the autophagy system. And Roberto Zonku, my valued colleague at Berkeley, was deeply involved in every aspect of the lysosomal complexes, and Sasha Martins in Vienna in much of the autophagy work and all of the reconstitution. And so what you see is our latest Zoom group meeting picture. So we're all missing seeing each other in present, and hopefully we will be back to happier times when the group looked more like that. So I hope you found some of that interesting, and I'm happy to take questions.
Thanks, James. Fantastic seminar. So any questions? Check.
Are you going to read the questions, or shall I? I saw Sean just posted one in the chat. How do you do this? Okay, Sean said the manner in which the folliculin FNIP fits into the crevice between Rag8a and C as part of its lysosome recruitment and GAP mechanism, along with the SLC38A9 and the amino acid sensing, is striking. Do you think that the GTPase regulation and amino acid sensing for C9orf72 and SMCR8 at lysosome will be similar? Are ARFs present at lysosomes and dimeric like the rags? So to answer the question backwards, there is absolutely no evidence that our ARFs function as dimers, and there's very, very little that they're at lysosomes. So the answers to that are probably no. Our working model is no. We were really very surprised and puzzled, and still are to some extent, at this finding that a lysosomal complex like C9orf72 has a famously non-lysosomal substrate, the ARF protein. So we have two models at present. One is that the C9 may be cycling on and off lysosomes, and it may actually be in its active form in the cytosol when it's off the lysosome. The other leading model in our minds is that the C9 is functioning at some type of contact site between the lysosome and an endosome, Golgi, or cytoskeleton. Good question.
So James, actually I have a very general question. So the ARF, GTPase, and the Rag, they play functions around the membrane, and since now the phase suppression is very popular, do you think is there any chance that they play functions by the phase suppression stuff?
Sorry, what kind of suppression? I didn't.
Phase suppression, lipid.
Phase separation. Yes, everyone wants everything to be phase separated. I keep telling my lab, if you just find that your paper will be guaranteed to get in Nature if you can find phase separation somewhere. And phase separation, I think, has been proposed by Nobuo Noda for yeast autophagy initiation, and there is a lot of IDR in this, and we think there is aggregation or assembly through the IDRs that may have some phase separation properties, but is probably occurring on a scale, a number of copies that's smaller. In one example where I think the phase separation is genuine, very well substantiated, is in P62 ubiquitin condensates as substrates for autophagy. I think that's, in my mind, the best substantiated example, if that answers your question.
Yep. All right, I think we're in a good time frame, and I know for the trainees, today is the trainee retreat, and a lot of you may have to attend the seminar at 2 p.m. But if you are available and if you want to join our roundtable, just follow the room linkage I sent to you yesterday. Thank you very much. Right, James, I think probably very good.
Right.
Then,
yeah, I once again probably gave you too much detail. I'll stop the screen share.
So yeah, I think you need to shift to the room, and I will see you after 15 minutes.
Right, I need to leave this link and